Okay, good afternoon, everybody. Welcome to the last colloquium of the year, this time organized by Coordination Dynamics. We have as our speaker today Daniel Ferris, Professor Daniel Ferris. He's the Robert W. Adenbaum Professor of Engineering Innovation at the University of Florida. So his research focuses on biomechanics and neural control of human locomotion. And I mostly know Prof. Ferris for his work on EEG during locomotion. So he's been a big inspiration for some of the studies that I've done. And I look very much forward to his talk. Daniel, the floor is all yours.
Thank you very much. I appreciate the invitation. So as you can see from my background, that's not real right now, but it's pretty close to what's out my window. I'm down in the Florida Keys and enjoying a little sunshine. Let me go ahead and share my screen. Okay, hopefully you guys can see and hear me okay, yes? I lost you. I don't hear you. I don't see you.
We can see and hear you okay.
Okay, so let's keep going. So I'm going to talk to you a little bit about how to use EEG to figure out what's going on in people's brains. And really, that wasn't my original intention when I started doing a number of research studies in grad school and even in as a faculty member. I was really intrigued by the human as a machine. It doesn't take much to watch an athletic event, whether you like American football or gymnastics or European football or lacrosse, basketball. The things that humans can accomplish is quite amazing in terms of their capabilities. And this always intrigued me growing up. Being an athlete myself, I played American football till injuries forced my retirement. And over the years, I started thinking about how do we study this behavior. And most of what I was drawn to was the mechanics, trying to understand the biomechanics of how the muscles produce the motion. But then I learned through grad school and my postdocs that the energetics are really important to shaping our motion patterns. So we think about how you move from point A to point B, and you usually do it with a mechanical pattern that minimizes the energy cost. And so you need to be able to sense what that energy cost is, and you need to be able to control it, the mechanics, in order to reduce the energy that the muscles are using. One of my friends, Art Kuo, calls it the principle of laziness. But there's lots of other things we do besides minimizing energetics. We're really good at responding to complicated and difficult environments. And here's a good example. I pulled this off of the internet of a gentleman who was bored with lockdown from COVID. You can see he's actually wearing a mask with it. And he was trying to challenge himself to go through a very extensive series of obstacles. And the point that I really just want to make is that we can't really build a replica of a human that can do these amazing agile feats. The level of control and the mechanics that humans can produce is quite amazing. And there's a lot about the balance, the mechanics, the control, the sensation that we don't understand. And there are lots of patient populations that have problems with their control and their balance, and trying to understand how people do it may actually help them in the long run. So when we think about the neural control of human locomotion, when we think about the neural control of locomotion, a lot of studies have been done on rodents, rats, mice, and they definitely can provide some insight into what's going on because you can become invasive. But when you stop and think about what humans are capable of doing, walking with your eyes closed, whistling a tune at the same time, texting on your phone, if you want, there's a lot of things that are extremely challenging that are hard to really understand unless you study them in humans. We usually divide up the neural control of human locomotion into three areas. Number one, the supraspinal areas, the brain, then the spinal neural networks, sometimes referred to as central pattern generators in a lot of non-human vertebrates, and then also the reflex pathways. And if you've been around the field long enough, you can remember back to when there was the presumption of Sherrington that the reflexes were the dominant pathway, and that the reflexes were super helpful and provided the activation for the muscles for taking step after step. And then when Sten Grillner and a number of his colleagues came along, they provided a lot of evidence that central pattern generators in the spinal cord were very important. And to a certain extent, I think people underestimated the involvement of the brain, the supraspinal areas. And over the last, I would say 20 years or so, we're getting a better appreciation of how much the brain is actually involved in the control of human locomotion. I like this image from Handbook of Clinical Neurology in 2018, because it really gives an idea of or appreciation of the many, many areas that seem to be directly involved with the control of human locomotion. It's not just the motor cortex or just the sensory cortex, but posterior parietal cortex, vestibular cortex, prefrontal cortex, brainstem, basal ganglia. There's an awful lot of areas that are involved in communicating and coordinating with each other for locomotion. And if it's a very simple walking at a constant speed on level, smooth terrain, many of these areas don't necessarily need to be contributing to the activation signals in the muscles with every step. But that doesn't mean that they're not monitoring what's going on and that they're going to take over should any perturbations come along. People have tried to understand human locomotion control from the brain by doing things like using functional magnetic resonance imaging of laying down a subject in a magnet and saying, imagine that you're walking. And that gets you a little bit, but you really can't provide a robust simulation of what it's really like walking just by imagining while you're laying down. It takes a lot of the information out. So even a few other research labs have tried variations where they've put a MRI compatible bicycle in the magnet, or they've actually used a giant cylindrical treadmill for subjects to take their steps as they try to look at the brain activity. But again, these do not take into account the things like the variability of the terrain, of the stepping, perturbations, vestibular feedback, visual flow. There's so many other things that normally contribute to the control of locomotion that really, if we want to understand it, we need to study humans while they're actually moving around in the real world. Think about going on a hike out in the mountainside. You have rocky terrain, you have changes in elevation, you have changes in incline and decline. If you're going through an urban environment, it's not unusual to navigate stairs and navigate other obstacles that are moving around you, like other people. So all of these things are how we normally move through the world. And so we need to sort of understand the brain dynamics while they're doing these tasks. So along these lines, in 2007, I began a quest to sort of figure out what's going on in the brain. And I took a sabbatical at UC San Diego to start using high density electroencephalography. This is me from my very first time being a subject trying to collect data while people were walking with high density EEG. And we've come a long way since then. And I need to be very thankful to Scott McCaig and Klaus Grauman. Some of you may know of them by their reputation, but they were very welcoming me into UC San Diego, where Klaus was a postdoc at the time, and he worked with me to help collect data and teach me what to do. And Scott mentored me on some of the aspects of the theory and helping design better experiments. And over the years, I've taken that knowledge and carried it forward. And one of the things that Scott is responsible for, along with some of his collaborators, is this software toolbox called EEG Lab, which I highly recommend. Has a lot of toolboxes that are set up for analyzing electroencephalography, and we still use it for analyzing most of our data. I also have to say that there are some wonderful beach volleyball courts in San Diego, so if you ever get down there to visit Scott or his lab, definitely stop by South Mission Beach. Highly recommend it. This path to sort of figure out what the brain is doing while you're walking around in the real world has required us to learn a very large number of different type of software processing approaches. Not only do you collect brain activity when you put electrodes on somebody's head, but you get motion artifacts from the electrodes moving around and people moving around. You get muscle artifacts because EMG electromyography, the electrical signals from the muscles from the face and the neck sometimes get picked up by electrodes on the scalp. You also get eye motion artifacts. When you look left, look right, look up, look down, when you blink, all of those things create electrical signals which are picked up. And over the years, a large number of scientists have come up with different ways to sort of process the data. And I'm not going to go into details about any of them. The take-home message that I sort of want to leave you with is there's no best method for processing EEG data. And it basically comes down to the fact that depending on what your experiment is, you may have different levels of rhythmic muscle activity or rhythmic eye activity or discrete muscle or discrete motion artifacts, different amounts of motion artifacts relative to brain. Because your experiment is uniquely different from other experiments, that means you have differing levels of eye, muscle, motion artifact to go along with the brain. And so you need to choose the processing method that seems to work best for your experiment. It's a little frustrating as a scientist because you'd like to be able to say, here's the best case pipeline. But when you look at different types of data, different pipelines perform with varying levels of success. And if you're interested in the idea, I recommend this paper by Sefadine in 2012. It does a really good job of showing how different pipelines work better or worse under certain circumstances. And so there really is not one best way to analyze all the data. So I show this picture of my teenage daughter because I want to remind you also that clean is relative. So this is a picture of Jasmine, my daughter's room, right after I had asked her, oh, did you clean up your room? And she said, yes, my room's clean. And I went and looked and this is what I saw. So I took the picture. To her, this is clean, right? And to me, maybe not the same level of cleanliness, but that sort of goes along with the data as well, is that you can clean it, but there's not an absolute gold standard clean for EEG data, and that you're going to have differing levels of artifacts still embedded within it. The general approach that we take is to combine high-density electroencephalography with independent component analysis, sometimes referred to as ICA. ICA is one of the blind source separation techniques that can solve the classic cocktail party problem. That's why I have the picture of the cocktail party. Imagine you're in a crowded room with 100 people and they're all talking and somebody says your name. You turn and look and you have no idea who said your name because there's so many people that are talking. But imagine for a second that you have 10 of your best friends that are all around the room and they all turned and looked at the same time. There are ways to take a vector approximation of where all of them are looking with their initial reaction, and you get a better approximation of where the voice came from. That's sort of how ICA works. So let's imagine for simplicity for a second that there are 4 electrodes on the scalp, shown here in red, and that there are 4 groups of neurons underneath them that are producing electrical activity. When I say groups, I don't mean individual neurons. I mean tens of thousands of neurons that are coordinated in their responses. And so they are producing what are known as far field potentials. That means a change in electrical potential that are large enough that it doesn't just dissipate as it progresses through tissue, but it can actually show up on the electrodes. If I look at those sensors, the EEG sensors, each one of those 4 red electrical sensors is going to pick up all of the sources. And so generally, the majority of people that do EEG analyze everything in EEG electrode space, or sometimes called channel space. What ICA allows us to do is create an unmixing matrix based on a large enough amount of data so that we can then reproduce those sources. And if you reproduce those sources, then you're actually looking at those brain areas as opposed to just the electrode locations. So if I start out and look at somebody walking on a treadmill in a lab, this is original data from a long time ago, of a subject walking on a treadmill. And then I do my ICA so that I get different sources. And I started with 256 electrodes. And out of those 256 electrodes, maybe 23 of them were bad, or 33 of them were bad. And I end up with 223 actual signals that I can analyze. And then after I get the independent components, I create an inverse head model based on the anatomy of their brain to say this is how electrical activity should propagate through the gray matter and white matter. You can actually create locations of all of those 223 independent components and show them and represent them with this electrical dipole. So all of these different colors are different sources of electrical activity that are propagating up to the electrodes. For an example, here's 3 of them that I'll pull out in red and green and blue, shown on a image of a human anatomy. And I can also look at a topographical plot. So what these are is, if you look at the top, there's a little nose and there's an ear on either side. And so you're looking down at the person's head. And it extends beyond the head because some of the electrodes go down along the side of the head and down by the neck and the forehead. And so this just represents how these different sources, here this green IC2, independent component 2, would primarily affect electrodes that are at the front of the head, shown by the red here. IC11 in blue would primarily affect the electrodes that are down at the far left side of the neck. And IC20 would affect primary electrodes that are up towards the front of the eyes. And in fact, not only do you get topographical plots and locations, but you get time series. So what we have here is versus time on the x-axis, the voltage changes in each of these components. And you can show that, for instance, IC2 and IC20 are really just reflecting horizontal and vertical eye movement. And so when you look up or you look down, you create an electrical potential from your eye socket that propagates and is picked up by electrodes. And then look at IC11, and what you see is not anything related to eye motion, but you see this rhythmic activity. It actually turns out the electrical activity from your heart propagates all the way up to your head and can be picked up by certain electrodes. ICA lets you pull that out. So data like this can make it clear that you're actually getting physiologically relevant sources that are somehow related to what's going on in the body. You can also pull out things like neck muscles, and when you localize them, they show up right in the outside part of the scalp where the neck muscles insert onto the skull. So after working with Scott McCaig and Klaus Grauman, we were able to take their approach back to our lab, and one of my grad students, Joe Gwynn, was able to start collecting data in our own lab. This is one of the original data collections in Michigan, where we're showing subjects an X and a plus and looking for a visual oddball response. So they're walking, they're running, and we were able to show that, yes, you can actually discriminate an oddball ERP, event-related potential, even while subjects are running on a treadmill. And so this led us to believe that we had something. There were a number of other
There appears to be, and now
I
I
will
see
take care of that little, hopefully it's gone. It actually shows up when you chat. So
okay.
I gotta take it out as soon as you chat.
Guys, gone now. Thank you.
Okay, so one of the downsides though is, having come from a field of biomechanics and not neuroscience and not EEG, is while these data were convincing to me, there were a number of people that were non-believers. They thought that mostly of what we had was motion artifacts and that it really wasn't brain activity, and that maybe we could pull out an event-related potential, but we really couldn't pull out anything meaningful out of the brain dynamics. So we took the discussion and we tried to figure out what could we do that would convince more people and ourselves that what we had was real. So we started building EEG head phantoms. And so what these are is essentially an electrical brain mounted within either dental plaster or ballistics gel mixed with salt that we could actually generate ground truth signals of brain activity and put it in different locations with different antennae, and then put electrodes on and actually test our pipelines and see if they were pulling out the ground truth. Because if we do this on a human, we really don't know what's going on in your brain. But if we do it on this head phantom, we know exactly what we're sending through the antenna in different locations. And so we can figure out if we can pull that out. Over the years, we've published a number of studies that have looked at the ICA validation and also connectivity measures validation. One of them by Stephen Peterson, who was a postdoc of mine who now works for Facebook. I'm sorry, he was a grad student of mine who now works for Facebook. He was testing out different connectivity measures. And he put a number of different signals into different locations within the electronic brain, using different levels of connectivity between brain sources. And so what you have here is basically just spectral power on the y-axis versus the frequency content, showing you examples of different types of signals he was able to pump through to the antenna with low, middle, and high frequency components. And then he used rather complex signals as well as rather simple signals, and tried a whole bunch of different connectivity measures. And lo and behold, we were able to find out that certain ones were the most robust to motion artifact and eye artifact and muscle artifact. And so in this paper, we were able to convince people that directed transfer function seems to be a relatively good connectivity measure for looking at how different brain areas are connected with each other from mobile EEG data. Our phantom heads have gotten much more complicated over the years. Here's our most recent one, who Ryan and Natalie, a couple of my postdocs, have deemed to be called Gary. And you can see we've gotten pretty complicated in producing different eye sources and muscle sources. But it actually works quite well. And we were a little worried about the hair effect because most of our early phantoms didn't have hair included. So we've taken it to another level by using various wigs made out of human hair and fake hair, and tested the relative effects of hair on the electrodes and the ability to pull out brain sources. One of the things that has helped us a lot in improving the signal quality is dual EEG electrode design, where we have our normal EEG electrode shown here in red, which has gel between it and the scalp. But we take a second EEG electrode and we invert it in blue. And so then we put a fake skin over the top. It's really carbon infused fabric over the top. And so when these two are mechanically coupled but electrically separated, this blue electrode is only picking up motion artifacts from the noise of moving around. And this normal EEG scalp electrode picks up both motion artifact as well as brain signals and muscle and eye artifacts. And so including this noise electrode signal into our ICA and using various techniques allows us to pull out the noise better because we have a cleaner representation of what the motion artifact looks like. So we've taken this dual EEG approach, put it on our phantom heads, put it on a motion platform. We took an IMU, an inertial measurement unit, put it on somebody's head, had them walk and run, measured how their head actually moves. Then we reproduce that motion on a motion platform while our phantom is bobbing up and down, and we send signals through the electrical brain. And if you look on the left series of data series, this is EEG signals or channels versus time. You can see how noisy it looks. On the right is our cleaned data from removing all the motion artifact that we're able to get from our dual electrodes. So we figured ways over the years to clean up our data and get a much better representation of brain dynamics. And in fact, when we try to reproduce random brain sources that we send through our Phantom, we actually do a pretty good job. So this top is our recovered black time series versus the original source being sent through a brain Phantom antenna. And you can see that in various different approaches, we get cross correlation R values greater than 0.9, showing that we're able to reproduce the actual brain sources. And so nowadays we have a number of studies that are continuing to use this. Here's an example from a recent paper by Andrew Norden, where we had obstacles thrown onto the treadmill that subjects had to respond to. And using this approach, we've actually finally, shown people that they're pretty strong believers at this point. I don't have many people saying that your data is junk. We'll find ways to make it better, but it definitely pays to be skeptical of your own data and try to think of ways to make it the quality of the data better. One of the things that long-term we're interested in is starting to figure out why people fall down. So many of you may know who this person is on the right. This is Shaquille O'Neal. This is back when he played for the Orlando Magic in Florida many, many, many years ago. On the left is a video of him falling down as an announcer now. So like all of us, we get a little bit older. We're not quite as athletic as we used to be. And in fact, you can do a search on the internet for Shaquille O'Neal falling down and get a lot of different videos. As we age, we tend to fall more. And so trying to figure out why people fall more is one of the goals of our lab. When we told IRBs that we're interested in people falling down, they had a lot of concerns. So we had to come up with a way to get people to lose their balance without getting hurt. And one of my grad students actually Antoinette Domingo came up with the idea of putting a balance beam on a treadmill. So what she was able to do is basically glue these 1-inch blocks on a treadmill so that they're walking on a balance beam forever. And you can see in this experiment, a data collection that Amy Sipp did in 2013, we published it, of subjects walking on this balance beam. And the advantage of using this balance beam is that they lose their balance and they step off onto the treadmill, but they're completely safe because all they're doing is just walking on a treadmill at a very slow speed then. But we can figure out when in their brain they actually realize they're losing their balance and they're going to actually step off. So what you're looking at here is data from 30 subjects worth of brain dynamics. Each up top are little circle or spheres represented by red or blue or yellow or orange or green or purple. And these are independent components from an individual subject related to loss of balance. And then you can see it from 3 different views up top, and down below you see the centroid or the mean location of all of the components that have been clustered together that are similar in electrical activity and location for all the subjects. And the first thing that I would like to sort of highlight is when you lose your balance, it's not just sensory motor cortex that responds. There's actually a lot of brain areas that show a burst of electrical activity or an inhibition of electrical activity related to the loss of balance. And then when we take the individual time series, we can create what are known as ERST, or event-related spectral perturbations. And so on the y-axis is the frequency content of our signal of a given cluster. On the x-axis is time. I've put little vertical lines related to beam contact, treadmill contact. So when their foot hits the balance beam, it's a BC. And when their foot makes contact with the treadmill, it's a TC. So they're losing their balance between the BC and the TC. And then showing you the cluster of right sensory motor cortex and left sensory motor cortex, the changes in spectral power in these 2 areas. What you can see is that the brain area that responds first to loss of balance is actually the left sensory motor cortex. And it actually comes on the minute that one of your feet hits the balance beam. Before the next foot is going to touch the treadmill, there's a huge burst in theta electrical activity, which indicates they know about a second before they lose their balance that they are not going to be able to keep their foot on the balance beam. There's data from bimanual upper limb coordination in fMRI and other means that suggests the left cortex is involved in a coordination between the left and the right. And these were all right-handed subjects, so I don't know if it would flip for left-handed subjects, but it's pretty in agreement with the literature that suggests if you do a bimanual upper limb coordination task, not only do you get activation in the left and the right sensory motor cortex, but somehow the left sensory motor is involved in that coordination. And it seems true whether you fall to the left or the right, or whether you step off with the left or the right. So this is actually in agreement with a lot of data from other labs that have shown if you lightly touch a surface with your hand, you're much better at maintaining balance than if you're just standing on the ground on one foot. And the idea is essentially if I have two flows of sensory information from my hand and my foot, then it's easier for me to calculate where my center of mass is. And the same is really true when you're walking on a balance beam, is if you have 2 feet on the ground, which is what happens when the BC occurs, both feet are touching the balance beam at this point, you're able to better calculate your center of mass location and velocity. And so you have a much better idea that I'm already outside the area that I can recover. So over the years, we've taken this paradigm and looked at other ways to figure out what is going on in the brain. Stephen Pearson, one of my grad students, was very curious about 2 different methods for training balance. Some people like to use physical perturbations, whether they push or pull your torso or whether they speed up or slow down the treadmill. And other people like to use visual perturbations, whether they occlude your vision or they rotate your vision. We really wanted to ask the question is, does your brain respond differently depending on a physical perturbation versus a visual perturbation? So what he did is got subjects to walk on a wide balance beam, so it was relatively easy to walk on it. And then either pulled at their waist one way or the other, or he had them wear a virtual reality immersive headset that had a camera on the front so that it actually filmed the real world, but it allowed us to manipulate the angle that it was being portrayed. He could enter in a short duration rotation of their view because he had a camera that was translating the real world view into a virtual headset. And so we could introduce rotations to the left or rotations to the right and see how they responded to the visual perturbation. And the basic question is does your brain really respond the same to all types of perturbations, or are different brain areas involved? So he had 30 subjects go through the experiment. Up top are, again, all the independent components that have been clustered together by their location and their spectral activity. Again, we put them into mean locations for all the subjects shown here. And again, perturbations influence a great deal of brain activity, not just occipital cortex, not just sensory motor cortex. And I want to point out these ERSPs, event-related spectral perturbations. So again, I have frequency on the y-axis, and I have time on the x-axis. Z occurs, the dashed line. The second dashed line at 0.5 is when the next visual perturbation goes away. So it goes back to normal view. And we wanted to say what brain areas respond to a visual perturbation. And as you might expect, the left occipital and the right occipital show strong spectral perturbations in the theta and then desynchronization. which means that a lot of different a lot of different neurons are doing independent calculations with that desynchronization in blue up in the beta range. But you see that there's very little activity in anterior cingulate, which is an error monitoring area of the brain, and very little changes in the left-right sensory motor cortex or supplementary motor cortex. So you're sensing a visual perturbation, and then the area of your brain that does integration of lots of different sensory flows shows a very strong response in posterior parietal cortex, but very little motor response. Compare that to a physical perturbation when you yank somebody at the waist. The supplementary motor, the left and the right sensory motor, the anterior parietal, and even the anterior cingulate all show a strong theta and even alpha synchronization shown in red. There's a little bit of right and left sensory motor beta desynchronization, but it's not as strong. But there's very little occipital and very little posterior parietal cortex. And so the take-home message that I would give you is that when you try to train somebody to walk better and improve their balance, the type of perturbation you give them affects vastly different areas of the brain. Visual perturbations affects this posterior parietal cortex, which is involved in integrating different sensory flows, but physical perturbations, not so much. Physical perturbations affect anterior cingulate, supplementary motor, left and right sensory motor, but not that those areas aren't really affected by the visual perturbations. So I would say long-term, if you're going to use a clinical intervention to try to help people balance better, know what their problem is, because different types of balance training are going to have different effect on the brain.
For the
Okay, so out in the real world, right, we normally come across a number of different types of obstacles, whether you're jumping over a log, you're walking on foot placements, you're in an urban environment and it's rained recently and you got to step over a puddle, or you're even a small child trying to put your feet in certain areas. We're actually quite good at this, right? It's not that difficult over the years. We learn to place our feet in exactly where we want them to maintain balance. And it can be actually quite complicated if you're trying to judge the unmoving obstacle coming at you. And not that I recommend anybody to try to jump over fast cars, but I point out that our brains are really good at timing when our body are going to reach obstacles and how we need to react to them. There's a lot of good research by Trevor Drew and Dan Marigold and colleagues that have looked at cats and invasive measurements of cats stepping over obstacles. Shown up above, a cat approaching an obstacle, they step over it. And it turns out one of the areas that is very much involved is posterior parietal cortex. And they show a ramp up of firing of individual neurons. So right now they're measuring spikes per second versus time as they get closer to this obstacle. And when you're over the obstacle is in the shaded area, you can see that there's an increase in firing of these individual neurons in the posterior parietal cortex, which seems to be timing of when you're going to reach the obstacle. And in a nice couple of review papers, they talk about how the posterior parietal cortex is really integrating information from visual flow, proprioceptive flow, and helping you navigate obstacles. So we were very curious if you'd see the same thing with humans running on a treadmill. So we had a number of subjects walk and run at various speeds, had little obstacles that came underneath the blind, and we wanted to see if we could show any evidence from posterior parietal cortex of when they identify the obstacle. So here's our event-related spectral perturbation. This is just a random one that I'm showing you because I want to orient you again. You have on the y-axis the frequency content in delta, theta, alpha, beta, gamma regions. The vertical lines represent, the first one is when the obstacle first comes underneath the sheet. The second one is when it's directly underneath the person. And then the end of the graph is when the obstacle goes off the end of the treadmill. And so we were curious, can we actually detect posterior parietal cortex that signifies when the obstacle is approaching? First question was, does this actually work for a range of speeds? And I show you ERSPs from 0.5 meters per second walking, 1.0, 1.5, 2 meters per second, and then running at 2 and 2.5 meters per second. And then again, the ERSP, you can see the vertical lines. And if you look left to right, the graphs are remarkably similar. So it seems like, yes, we're getting good signal quality at a range of different speeds. It looks like the width of your spectral synchronization, this red bar, is thicker, but it's actually not thicker. It's just because you're moving at a faster speed, the amount of time from when the object appears to when the object is gone is much shorter. So we've stretched out the x-axis here compared to here, but actually you see the same amount of spectral synchronization that occurs across delta, theta, and alpha for all of the speeds. And this is in the premotor, which was surprising to us that it would be so strong. But in fact, we found 3 different brain areas that were very responsive to the obstacle. We found premotor cortex, primary motor cortex, and posterior parietal cortex. We're all showing activation synchronization to the obstacle. And if we look at the timing of them, the premotor and the primary motor didn't really change the activation onset for different walking speeds. It was all about the same, somewhere around about 150 milliseconds, regardless of speed. But if you look at the posterior parietal cortex, it came on later and later at slower speeds. The activation onset took longer until it was basically coming on right at the step before you would go over. And so the premotor and primary motor cortex respond to presence of the obstacle on the treadmill. Immediately it comes on. The posterior parietal cortex on the other side comes on in the step before you step over it. So it was very much showing maximum activation latency when you're about ready to step over. And at the very fast speeds, it was actually as soon as the object came on, because your next step was going over the obstacle. We're still working on better ways to clean up our data. Ryan Downey is a research faculty member in my lab who's come up with a way to do the cleaning in real time, which is really cool to us. So we're still using the dual electrodes, but instead of post-processing all of the cleaning to try to get rid of the motion artifact, he's using a new software algorithm that he's come up with that's based on CCA. And that allows us, CCA is also known as chemical correlation analysis. The nice thing about it is it's computationally quick and inexpensive. And so we can actually clean up our data quite quickly. And so what this shows you, for an example, is a subject walking on uneven terrain. This is the raw EEG. This is the projected noise components in real time. This is the raw noise that's been pulled out of the noise components. And then this is the cleaned up EEG. And so if you compare the upper left with the upper right, you can see that we're able to get much cleaner EEG using this in real time, which is nice because if you think about, could we actually use EEG signals as a way to create a brain-computer interface or to signal information to your mobile phone, your smartphone about what's going on in the brain? We're finding ways that will allow us to clean it up without having to do a great deal of post-processing. So along those lines, we have a clinical trial ongoing right now of older, healthy adults walking on a range of different types of terrain. Here's our latest version of the uneven terrain treadmill. These are just hard foam pucks that are Velcroed to the surface. So we can actually remove the foam pucks and then put different ones down. So we can vary the level of unevenness. They can be higher, they can be more variable. And we're very curious as to how the brain is responding to navigating these uneven terrain, and then how do older adults in particular respond to navigating the uneven terrain. We had another study that was funded by a National Science Foundation to try to take this to actual tennis play. Going back to my initial interest, you remember back at the beginning of the talk, I told you I was very interested in sports and the human machine. I would love to be able to monitor brain dynamics during tennis. This is actually a picture of my wife, Rachel Seidler, as an experimental subject in the tennis study. Unfortunately, what we've learned over the months of trying to collect data is that all of the neck motion of looking up to serve and twisting your head left and right and the huge amounts of shoulder activity is creating more muscle activity and strain on the cables than we can correct for. So, you hear a lot about research successes in talks. What I'm going to tell you is a little bit of a failure is we weren't able to get clean data from tennis. Even though we thought we had come up with a really good design, we're just getting so much muscle and motion artifacts that it's dominating the signal. So we've reoriented this study, and we're actually showing that with table tennis, which is not requiring nearly the same amount of muscle activity, because you're not taking as broad strokes, the bracket isn't as large, you're not hitting the ball as hard. We're actually doing a pretty good job. So Amanda Studnicki is now taking on this table tennis project, and we're able to figure out what's going on in the brain while they're intercepting the ball and tracking the ball when they make mistakes. So I hope in future studies to give updates on that project. We know there are a lot of companies out there that are trying to come up with mobile EEG systems. Here's just a few of them that are pictured. It's definitely a technological boom right now, and not all of it is really geared towards researchers at universities or even clinicians. A lot of people would like to be able to have these mobile EEG systems set up for video games so that you can know when people are bored or excited or overstimulated and have it change what's going on in the video game. Or it can monitor your brain dynamics during a fatiguing, long occupational task and give information back to you to warn you about certain things, like when you're driving and becoming fatigued. There's also a whole field of neuromarketing where you take individuals and have them go through a store or a mall, and they observe what's going on, and you use that electrical activity to figure out how to better design the mall and the displays so that you can sell more products. Long term, I'm most interested in the clinical aspects. I would love to be able to actually monitor people's brain activity while they're actually doing something like playing American football and they get a concussion. What's actually going on in their brain, and how do you figure out how the electrical activity is affected? How do we better do rehabilitation amongst elderly? How do we build brain-computer interfaces for things like exoskeletons? Here's a picture of a project out of the European Union on the Mind Walker. And I think these will be possible in the near future, in the next 10, 15 years. There's a lot of work that still needs to be done, but we're making progress. And I definitely give accolades to the groups that are doing this. As a beach volleyball player, I'd love to get to the point where we can monitor brain dynamics while you're doing very active sports like playing beach volleyball, or using it to train individuals with virtual reality and monitor brain activity. I think there's a whole range of possibilities out there that are going to become viable in the near future. When we think about things like brain-computer interfaces, this is an example of a brain-computer interface that's used to just move a cursor on a screen with the idea that eventually it can help people that are paralyzed control a robotic arm. If you know your Marvel Cinematic Universe, then you know about things like Iron Man's helmet that allows him to interact with his robotic exoskeleton just by thinking about it, which really is a brain-computer interface that uses EEG. And then Professor Xavier and his Cerebro is another way to interface with the computer for a person with a disability to perform tasks that they normally couldn't. I think all of these really are the future. I want to give a few shout outs to some of the people that have helped do this along the years. Eva, Klaus, Natalie, Christine, Andrew, Scott, Joe, Troy, Stephen, Anderson, Julia, Helen, Amy, Ryan, all of them have been amazing colleagues to work with. And I also want to thank my funding sources, NIH, NSF, ONR, ARL. Thank you for listening.
Thank you, Daniel. Are there any questions? I see Andreas opening up his microphone, so I'm sure he's going to.
I do have a question. I don't know whether I'm the first to ask.
Just go ahead.
Well, thanks very much for the very nice overview. I mean, I'm very happy to see that you also struggled with getting the tennis stroke on EEG. We tried our best with basketball and switched over to darts afterwards, which is less demanding, at least when it comes to muscle activity. I'm also happy to see that you're still trying to improve your artifact removal. I think that is really the strongest part over the years. Yet I'm a little bit puzzled, to be honest. I mean, if I look at the one study that you showed, I think it was by Nordin in 2018-19, that was about walking, and you showed this premotor area activity. I mean, when I look at these, I mean, you went to really, really low frequency components up to the delta frequency, which I personally would consider to be still artifacts. I mean, I guess you have a different opinion on this one, but also the sources that you infer by your ICA or CCA approach are very, very deep in the brain. I wouldn't call these premotor areas, to be honest, but I mean, also there. I mean, how sure are you that I mean, the entire pipeline really, really recovers proper location and the proper frequencies? I mean, the phantoms are one thing, but these high impact forces during walking, running might be something different.
Right. And so I think it always pays to be skeptical, right? So what can you possibly do to convince yourself that what you have is real and test the hypothesis? Okay, it really is artifact. How do we convince ourselves that it's not? And so if you go with the assumption, if it's artifact, then I should see it increased or decreased in certain situations. And to me, the electrical head phantom has been our biggest argument, because we can introduce the electrical signals in lots of different brain areas and make them complex. We can make them simple, and then we can add in motion artifact or not add in motion artifact. And if we're getting the same signal recovered from the electrical brain, regardless of the motion artifact, then the motion artifact is not bleeding into the location. As opposed to one of your specific questions is how confident are we on the exact location? I would say that there's a lot of studies that have looked at different ways to measure EEG location using the techniques we have. And at best, I would say it's 1 centimeter. So, you know, it's never going to be fMRI where you get millimeter accuracy. I would say that if we're showing a source in a given location, I'm pretty confident it's within 2 centimeters. Now that's a big area. There are a lot of studies that suggest, and best case scenarios, you're going to get to 1 centimeter location. And there's a huge literature, and some of the most recent stuff, Martin Siebers, for instance, used patients with deep brain stimulators so that they knew the location in deep brain locations that they were stimulating. And then they used EEG to do source localization, and they got within 1 centimeter in these deep areas. So, you know, that's a good example of, in humans with all the variability of gray matter and white matter, you are specifically using the human as an electrical head phantom by stimulating certain areas, and yet you're still able to show that you're within 1 centimeter of location using these same techniques. I think that's a pretty strong, convincing argument, at least to me, that even as we get deeper and deeper, it's not too far off what we think we're finding in terms of the location. There are a lot of older studies, which I can't believe they ever let them do, where they took recent cadaver heads and they put electrodes in them and they stimulated and they used these techniques and they tried to figure out, and even those again, are about 1 to 1.5 centimeters in localization error. So again, I don't think we're ever going to get to millimeter accuracy, but I'm willing to bet we're within 2 centimeters.
I mean, pre-motor area is not a millimeter accuracy, but these are really deep in the brain. But anyways, I mean, I fully agree. I mean, but you triggered away my follow-up question, if I may. You mentioned this 1 centimeter resolution. I mean, we have a set of simulation studies showing exactly these as a boundary. I mean, if you take 64, 128, or 256 electrodes, it all ends up, even if you're on the cortical level, you go down as much as to 1 centimeter resolution, even in sensory areas. But one essential ingredient in order to achieve that in our studies was that as soon as you have a more complicated spatial correlation between source or sensors, then you quite surely have to apply spatial pre-widening. And your ICA, CCA approaches strongly rely on temporal pre-widening, which is an add-on that you have to do all the time anyways. I mean, we all agree on this one. I mean, ICA, we use ICA a lot for artifact removal. But when it comes to spatial pre-widening, this is not so favorable. Not people use it, though it is pretty much the same technique. Did you ever try to do your phantom with correlated, I mean, in highly nonlinear correlated source activity and see how good your CCA inference would be?
So yes and no. We have tried highly non-correlated signals in the phantom, but we haven't taken it to the level that we analyze. So the best examples I can give you are with Stephen Peterson's Journal of Neural Engineering paper, where he looked at the connectivity measures. In some of that, we actually tried a whole bunch of different types of signals and trying to figure out the relative success of different approaches. And we haven't looked at it in the approach that you normally take, but it would be something we could do because we still have the data. Another thing in response to your earlier question, yeah, I'm not a big fan of looking at delta and making conclusions on delta because I agree it is quite low. I was a little skeptical of theta, but the theta activity really is strong for certain types of perturbations. So it really comes up very robustly when you start looking at sensory motor perturbations that are affecting overall balance. So some of the examples are the visual perturbations where you get this strong theta synchronization followed by beta desynchronization. We're finding that same pattern over and over again. And in many instances where it's purely seated subjects and everything is based off of cognitive and there's no motor component. So I'm often very skeptical of data in terms of making a conclusion on it. But if I get up to theta and it's a very robust result that across multiple types of behaviors, then I think it's got a stronger theoretical basis.
Thanks.
Other questions? I thought I saw Jeroen raise a hand.
Yes. I have a question, and I'm far from being an expert in EEG, even though I've never measured EEG, but I was wondering something that you mentioned in the beginning, that one of your independent components, that was your correspondent to your ECG or your heart. So apparently you localized in your method, your heart being in the brain. So apparently that's wrong. And so I was wondering.
No, so let me explain. I should have done. I usually do when I talk about it. So imagine a 15th century map of Europe, right? Once you get off the edge of the map, it says there be dragons here, and we don't know what's there. That essentially is where the heart showed up. It was on the very border of our map as outside of the head, but it didn't know where to put it, so it put it on the very edge of the map. And that's, you know, in reality, it's putting it in the closest place that the electrodes would measure, down in the neck where our map ends.
Okay, thank you for clarifying.
other questions? So I had a more general question in the beginning, which as you know, I've done some EEG studies as well, and we're working on it as well now. And you said, so there's not one best way to analyze EEG. And so in a way, this is a problem, right? Because we have so many degrees of freedom, and like, as researchers, we can do so many things. What do you think of this? Do you see a role for pre-registration here, or do you say, well, we should just be honest and report what we did, or you should always try it in 3 or 4 different ways, and only if it holds up in 3 or 4 different ways, you like trust what you're doing?
So number one, I'm not against pre-registration. I just think in terms of the things that we can do, it's lowered down the priority list for me. Much more higher priority is sharing data. So I think if we do things our way and we get a conclusion and then we put the data up, if there's anybody that has a problem with the way we analyze it, let them analyze it themselves from the raw data on and see if the result shows. So as I'm editor-in-chief of IEEE Transactions and Neural Systems and Rehab Engineering, which is an international journal of primarily engineers, there are so many engineers around the world looking for data sets that they don't have capabilities to collect themselves, that if we can put our data from EEG studies up on data sharing source websites, there will be people that will analyze it. And I think that's going to be more helpful to get things moving, to try to figure out what are the best ways to process and pipeline data. Is the more we can get different labs doing it. Like, I'm a big fan of the, there's a push with fMRI and EEG to have many labs around the world analyze, classic papers and see if they find the same outcome. And I think it's the same way, is we have to show that there's reproducible effect. That's one of the reasons why we've moved number one to going to 30 subjects minimum for EEG studies, because I think it's much more robust over time. And if we find the data in 10 subjects and then another 10 and another 10, and whether you analyze them all independently or all together, you get the same result, that's a clear indicator to us that you're finding something that's going to be universal to many other subjects. I think sharing the data, posting the data, and letting other people have access to it is a good thing. Just recently, another lab published a paper off of one of our data sets, and I was so happy to see it, right? But I think that's going to push farther than pre-registration. I don't think pre-registration is bad. It's just, I feel like so many times going into a study, we thought we'd find one thing, and yeah, we didn't find it, and I could publish it, but it's much better to say we didn't find it, but we found this, and this is better. And I think let's not blind ourselves to only looking at one question, but let's try to be as forthcoming about our data as we can.
Well, I mean, this doesn't speak against pre-registration. I mean, you can pre-register your experiment, collect the data. If you don't find things, or even if you find things, you make your data publicly available and let others take them for assessment.
I agree, and again, I'm not against it. I just feel like I think the problem that pre-registration solves is much smaller than the problem that sharing data would solve.
True. But I mean, one point on this one. I know that Jeroen wants to have the finishing in time, but I mean, let me take the opportunity. Do you know of anybody who used your data with your ICA source localization approach and tried to compare it with beamformers?
I'm not going to speak for him, but I'm pretty sure that Jose Contreras Fidel at University of Houston has done this. So in chatting with him at a conference, he used to use beamformers all the time. So if you look at his older publications, that was the way to go. And he and I chatted about using ICA. He then said he would do an analysis to compare, to convince himself what he thought of his data. And I haven't talked to him at a meeting since, but he's totally switched to ICA now. So I don't know. I would love to see him at a conference, but with COVID, it's shut down. So I don't know why he convinced himself to stop doing beamformers.
Well, I mean, to be honest, ICA is faster than beamforming, I suppose, but I mean, that also helps.
No, it's again one of those things that would be nice to be done, and hopefully, as we put more and more of our data online, then people can try a number of different approaches and see if there are differing opinions.
Marcy?
Thanks, Daniel, for the nice presentation. I have a quick question about the data that you show for the balance on the treadmill, the balance beam. And the EEG that you recorded, was it with the electrode that you showed with the removal of the artifact, with the noise?
No, it actually, that data was before we had come up with the design, but truth be said, if you're moving very slow, so the speed of the treadmill was like 0.3 meters per second, which is very slow. There's really not that much movement artifact when you're walking on a balance beam at 0.3 meters per second. So if you're going to do slow walking speeds, I don't really think you need to use a dual electrode approach.
Yeah. And for this balance study, the subjects had their arm swing, or I don't remember from the presentation, or arm crossed.
So in the initial study, we had arms crossed because we wanted to make the task more difficult.
Yeah.
And the other thing that we wanted to do is prevent people swinging their arms to having a long delayed touching of the treadmill. So a lot of times when you're walking with your arms free, you can use the arms to create angular momentum that keep you from losing your balance. And so we wanted a cleaner, hey, I realize I'm losing my balance and now I've touched the treadmill belt. And by preventing arm swing, it did a more reduced variability from event to event to event.
So at the time that they were out of from the beam to the treadmill, there were no arm movement, basically.
Right, correct.
But there is still a leakage, right? I know that, for example, if you don't move your arm, there is a still, but yeah, anyway.
We have other experiments that are doing arm movement now. And the main reason is we're trying to use the balance beam walking and visual perturbations as a way to train older subjects to improve their balance. And so it's a lot easier to make them feel comfortable if they can move their arms. And so we have some ongoing experiments with arms free. And as far as I can tell so far, it's not changing anything. It just makes the task a little easier and it gives more variability from loss of balance to loss of balance.
Thank you very much.
You're welcome.
Okay. Wait, no, everybody switch. I see that Jaap has one more question. I'm going to allow that as the last question. Jaap, go ahead.
Okay, thanks, Sjoerd. Thanks, Daniel, for a very nice overview. I was intrigued by this study on the balancing as well. So you link the changes in brain activity that you see with the realization that you're losing balance, right? Now, the consequence of losing balance, touching the treadmill, is quite late in this kind of paradigm. So I was thinking whether you could maybe, well, whether you thought about, let's say, an experimental design that might be stronger in this respect. And I was thinking back of an experiment that James Aston Miller talked about many years ago, and he had people just balance on a chair, so on the two hind legs of a chair, and then when they lose balance, of course, they have to grab the table to prevent fall, and there you can have a very short delay between, let's say, becoming aware that you're losing balance and making it, or actually performing the act. And he was able to relate that to the statistics of the sway pattern before that could predict when they made that switch.
Yeah, so I'm actually very familiar with those studies. So his PhD student at the time was Alea Ahmed,
right,
who was at Michigan, and I was a faculty member at Michigan, and Alea was in my classes, and so I was on her dissertation committee. Huh. So I know those, and we've thought about doing it. And it just comes down to limited people and limited effort. Right now, we're trying to push forward to actual clinical interventions, trying to use, perturbations as a way to train individuals. But I would love to do that experiment. You're right, it would be a much cleaner design of being on the chair and leaning back, and the instant that you detect it. And my guess is, if you collected data from that experiment from the same time as you did balance beam walking and put all of the data in for the ICA, you would show the overlap in the brain areas that are showing the same result.
And it could be that actually that the ability to detect an impending loss of balance could be an important predictor of falls, and maybe extrainable as well.
Yeah, so one of the reasons we actually got started on this is one of my students asked the question, is it when older people fall, is it that they don't detect they're losing their balance? They'd have sensory problems? Is it that they're not doing the calculations fast enough in the brain that let them realize how to respond and activate their muscles? Or is it that they tell the muscles to respond and that they just don't have the biomechanical strength and power to do it? And we were hoping that by following older subjects' loss of balance, we could actually figure out, compared to young, are they detecting it at the same speed, or are they detecting it and then they just can't figure out how to respond? Or is it they send the signals and then they just don't respond, right? Like, because you could hopefully break it down into those different areas.
Right.
And we still hope to do that study someday.
Thanks.
Okay, Daniel, thank you once again.